Okay.
Hello everyone. Hello everyone. We welcome you to our webinar titled Applying In Silico Metabolism Modules to Help Characterize Drug Candidate ADMET Properties. Today we have with us Dr. Mike Lawless, who is the team leader of Cheminformatics Study Team. Michael will be covering the major portion of the talk today. Myself, Anu Sharma, I am the Senior Manager for Sales and Marketing. I will be giving you a brief introduction to our product portfolio. Then on the panel, we have Dr. Bob Clark, the Director of Life Sciences, and Dr. Robert Frekovich, who is the Team Leader of ADMET Predictor. They will be taking any questions towards the end of the program. So please put in your questions in the question box on your control panel, and we will be answering the questions in the order that they were received. So before we begin, let me give you a brief introduction of the company. We were founded in 1996, headquartered in Lancaster, California. Ours is a publicly traded company with 25 employees. We are devoted to developing modeling and simulation software, which is used for the analysis of data mining, estimation of ADMET properties, and simulation of in vivo absorption, pharmacokinetics, pharmacodynamics, PDDIs. We also offer services for the simulation and cheminformatics software. On the right-hand side of the panel, you see is our product portfolio. Gastroplus is primarily a product for biologists. ADMET Predictor is used by the biologists as well as the chemists. MedChem Studio is a product for medicinal chemists, and DDD Plus is a product for formulation scientists. MedChem Designer is our free tool which is offered to anyone needing to draw structures. As you see, that Simulation Plus offers value at various stages of the drug discovery programs. ADMET Predictor is a product that is used in the earlier phases of the drug discovery to the development stages, and it is used for the simulation and estimation of physical, chemical, and biopharmaceutical properties, metabolism, and toxicity values as well. MedChem Studio is a product used for data mining and lead identification and de novo drug designing. MedChem Designer, as we said, was a tool for anyone needing to draw the structures. Gastroplus is a tool that comes in the development and in the clinical stages as well, and it's used for the simulation of intravenous, oral, ocular, intranasal, pulmonary absorption, pharmacokinetics, pharmacodynamics in humans and animals, and to predict DDDI in population outcomes. The DDD Plus is the first and the only simulation tools used for in vitro resolution experiments of APIs. Next month, we will be releasing a number of new product features and updates. And since this talk is focused on the Metabolism Module, which is a part of ADMET Predictor, I will be covering the new features in the upcoming ADMET Predictor 6.5. So as you see, ADMET Predictor 6.5 will be covering new features and will be bringing new changes in the general program applications, ADMET Predictor modules, and the graphic user interface. The first and the foremost thing, the good news, is that the ADMET Predictor 6.5 will be using the FlexNet Manager. This is a server-based license management system used in the industry for past 2 decades, and most of our customers are familiar with it. So in short, it means that you do not have to make phone calls to us to activate your license. Second feature is that we will be bringing in prediction confidence. So you will be able to predict with confidence. In other words, for selective classification models, you can see the probability of the correctness of the prediction in the spreadsheet as shown here. We are introducing several new predictive models. One of them is the Henry's Law constant used for the air-water partition coefficient. We also are bringing in another predictive models for skin permeability. We are also updating our Ames mutagenicity models. There are 10 rebit models for Ames mutagenicity. We've also improved the minor 3D graphing. And we are also bringing in new site-specific CYP-mediated models, the details of which will be covered in Michael's talk. So with this, I would like to hand over the presentation to Michael to go over the details of the metabolism module. Over to you, Michael.
Thank you very much, Anu. Good morning and welcome to the webinar. This next slide is simply an overview of the talk. So first I'll simply discuss drug metabolism in general, talk about cytochrome P450 metabolism and inhibition. Then I'll show kind of a flow diagram of the models in the Metabolism Module of ADMET Predictor. Then I'll discuss the classification models within the metabolism model. The two models are CYP inhibition and substrate classification models. In the next section, I'll talk about sites of metabolism. So we have, if a molecule is a substrate for CYP, then we also predict the sites of metabolism and then also can display the metabolites within our MedChem Designer program. The next section I'll discuss our new kinetic property predictions for CYPs, which will assign kinetic properties to particular atoms that are from the sites of metabolism. And then in the last section, briefly discuss our UGT models. UGT is involved in phase 2 metabolism of drugs. This slide is an illustration of metabolism of an orally dosed drug. So let's say the drug is delivered or created in pill form, would be ingested, dissolve in the stomach, and then enter the gut wall in the intestine. The gut wall is made up of enterocyte cells. The enterocyte cells contain cytochrome P450 and UGT. So this is the first opportunity, or one of the opportunities where a compound can be metabolized. Molecules will then pass through the gut wall into the portal vein, where they will be delivered into the liver. The liver contains a large number of different isoforms of cytochrome P450 along with UGT. So this is another area, or the largest area, where compounds are metabolized, especially for CYPs. UGT adds a glucuronic acid to the molecule and, of course, makes it much more hydrophilic and easier to eliminate. In 2004, in Drug Metabolism and Disposition, this publication illustrated the enzymes metabolizing the top 200 drugs. So here you can see that about 3/4 of the compounds are, about 3/4 of the metabolism is due to cytochrome P450, a little bit lesser extent from UGTs, and a smaller extent from esterases. For the cytochrome P450, the 5 major CYPs are 1A2, 2C9, 2C19, 2D6, and then 3A4 and 3A5, with lower contributions from 2E1 and 2B6. And we have certain models that will encompass all of these isoforms of cytochrome P450. This next slide is just an illustration of a cytochrome P450 reaction. So this is a crystal structure of cytochrome P450 2D6. So a molecule such as propranolol would bind to the receptor. Oxygen would be transferred from the heme group onto the 4-hydroxy position of the propranolol to create 4-hydroxypropranolol. So typically, one of the functions of CYPs, the molecule, once it's metabolized, ends up being more hydrophilic and easier to eliminate. So in this case, our predicted log P for propranolol is 2.96. The 4-hydroxypropranolol is lowered to 2.19. In the case of inhibition, such as a compound such as sincalcet, which is a strong 2D6 inhibitor with Ki of 87 nanomolar, it would bind into the receptor and then prevent a compound such as propranolol from being metabolized by cytochrome P450 2D6. So what happens in this case? What would happen is that this molecule is no longer being metabolized, so it tends up to build up and increase the systemic exposure and can cause maybe problems with toxicity of that. So this is one reason why the FDA emphasizes experiments to look at drug-drug interactions and inhibition of CYPs. This slide is an overview flowchart of our metabolism module. We have classification models for CYP inhibition of the 5 major isoforms of cytochrome P450. Then we have 9 classification models for is the molecule a substrate of a certain CYP or not. And here we have the 5 major CYPs along with 2A6, 2B6, 2C8, and 2E1. If a molecule is a substrate of a certain CYP, then we will also predict what sites on that molecule are metabolized, and then these metabolites can actually be visualized with MedChem Designer. Our new models include kinetic parameters for Km, Vmax, and clearance on an atomistic level. So for each one of the sites metabolism, we will also predict Km, Vmax, and clearance. And then finally, we also predict, we have classification models for 9 isoforms of UGT. So these are, classification models. Is this compound a substrate for the various isoforms of UGT? This next slide is an overview of our model building procedure. So we start with structures and experimental data. We take a lot of pain to make sure that the structures are correct and the experimental data results are pertinent to the model. So we start out, we would have structures as rows, and then the biological activity or the property value we're trying to predict associated with the structures. The next thing we do is compute more than 300 descriptors. And now you can kind of imagine this as a table where the rows are the molecules and then the columns are the various descriptors. We remove low variant and correlated descriptors, and then we go on to create a test set. And this is truly an external test set. It's never used in the model building procedure, and it's only used to, once the models are built, we predict the activities of the test set. So the remaining molecules go into a training set, and these are the next a priori. We don't know what number of neurons and what number of descriptors create the best model. So what we do is experiment and create a large number of models with different architectures, i.e. different number of neurons and descriptors. We use sensitivity analysis to determine what descriptors will create the best models given a certain number of descriptors that are specified in this step. Then the training set is used to develop artificial neural network ensemble models. The models are then applied to the test set, and then we can visualize the statistics in the grid down here. And what you want to do is avoid overtraining your model. So if you look at a model and you have very good statistics for the training set, but very poor statistics for the test set, then that would indicate you're overtraining the model and you're kind of memorizing the results in the training set, and it's hard to apply it to molecules outside that training set. Our ensembles are made up of 33 artificial neural network ensembles. In the case of a regression model, the final prediction for the molecule would be the average of the 33 artificial neural networks. This illustrates the 2 types of models that can be built in ADMET Predictor. One is a classification model and the other is a regression model. So in a classification model, we have a binary input of the experimental results. In this case, we're looking at non-toxic and toxic compounds. So this would be your negatives, and the toxic compounds would be the positives. Then the model is going to predict the toxicity of these compounds. If in the lower left-hand corner, these are the percent of compounds that we correctly predicted, they are negatives, and we correctly predicted them as negatives. In the upper right-hand corner, we have the true positives. These are the compounds we predicted to be toxic, and in fact, they were toxic. Then along the other corners, we have the incorrect predictions, so false positives and false negatives here. And on the next slide, I'll talk about the model statistics. Over on the right-hand side, we have a regression model. This is for our log P model. So we have the observed values and the predicted values. This is a continuous a variable model, in other words. And we get excellent statistics in both the test set and the training set with R-squareds above 0.97 for both of those. So when we build our models, we pay particular attention to sensitivity and specificity. And the reason is that if you have a model, and I'll show an example of this later, where you have 90% of your compounds are observed negatives and 10% of your positives, and then you have 10% of your positives, well, you could just predict all the compounds to be negative, and you'd be right 90% of the time. But your specificity is a measure of the true negatives over all these will be 1, whereas your sensitivity will be 0 in that case, because you'll have all the positives incorrect but all the negatives correct. So that would be an imbalance. So we seek a balance in the sensitivity and the specificity, particularly when you're dealing with imbalanced data sets. Okay, this next slide, this comes from a website from the FDA and just iterates the how important drug-drug interactions are, leading to changes in systemic exposure and potentially adverse drug reactions or loss of efficacy. And they say that we should pay particular attention to CYP-based drug-drug interactions. This next slide illustrates our performance results on the inhibition classification models for cytochrome P450. So we have 5 models of general inhibition of CYP, and then 2 specific models for 3A4, in one case where midazolam is the substrate, and in the other case where testosterone is the substrate. The data for these models varies. So for CYP1A, we have sufficient data with measured KIs for this particular isoform, and we set a threshold of 30 micromolar. So any compound that has a KI lower than 30 micromolar is labeled an inhibitor. Anything above 30 micromolar is a non-inhibitor. For some of the other models, the exact thresholds cannot be specified because the public databases only contain binary indications. 2C19 was a little bit problematic also because there's very little information on non-inhibitors. So we accepted non-inhibitors that were non-substrates for 2C19 and were not previously identified as inhibitors of that. So if you look at the overall statistics, they're very good overall. Everything above the lowest one here is 73%. We're reaching 95% concordance on the training set for 1A2. And then similarly good results for the test set. Now I'm going to pop into ADMET Predictor and kind of demonstrate the models there. So this is ADMET Predictor, and I've read in a set of 24 antidepressant molecules, and then to calculate the properties, I simply go to the Calculate pull-down menu and click on Calculate ADMET Properties. Then I click on the Run options, and this very quickly calculates all the descriptors for these 24 molecules, and then uses those, the models use those descriptors to create the predictions. We also, in that amount of time, have created over 140 predictions for these various molecules. The visualization of the models in the spreadsheet can be specified via the tabs at the bottom of the window. So initially we show all of the models and descriptors in the All tab. The user inputs will only show what's input with the file in this case. We have physical, chem, and biopharmaceutical properties here. And then I'm just going to click on the Metabolism tab here. So you see we start with metabolism of 1A2 inhibition. So this is a classification model for 1A2 inhibition. Then we have whether it's a substrate, etc. And then various other isoforms. And then down at the very end, we have our UGT models here. You can also customize these buttons and therefore customize the view in the tab. So what I'm going to go and specifically look at CYP inhibition for these molecules. So here are the 5 CYP models, classification models, again, yes or no for the various isoforms of cytochrome P450. So I'm going to go down to fluoxetine, and as you see, all of our predictions here is that fluoxetine is an inhibitor of these 5 isoforms of cytochrome P450. And if you look up this compound in the drug bank, it does show that fluoxetine is a substrate and an inhibitor of all of these CYPs. So one thing I can do, another thing I can do is visualize the molecule in MedChem Designer. So I simply hold down the Shift button and click on the image of the molecule, and that pops it into MedChem Designer. I can then push on the AP button to calculate the descriptors and the properties for this molecule. And as you see here again, I've ordered the table down here and am showing the inhibition of 1A2 and 2D6, which are both positive here. All right, so let's say I want to try and modify this molecule to come up with a new molecule that is predicted not to inhibit these 2 enzymes. I simply copy the structure and paste it. And what I'm going to do is modify the phenyl ring down here to a pyridine ring by simply converting one of the carbons into a nitrogen. Again, I push the AP button, it calculates the models, and now we're predicting a non-substrate for 2D6. That's very good, but we still have 1A inhibition. So again, I'm going to copy this molecule just so I can save the old molecule results. So I copy and paste it. And then what I'm going to do in this case is remove these fluorines. So convert the trifluoromethyl group to just simply a methyl group. Again, push on ADMET Predictor to calculate the properties for this molecule. And as you see now, we're predicting that this is a non-substrate of 1A2. That's very good, but the methyl group is not electron withdrawing. And maybe for the pharmacophore of this, they put a CF3 group there for electron withdrawing. So here I'm going to convert the methyl group into just a fluorine atom, push AP, and then see what happened to the model. Again, it's still, now it rearranged it, but here's the results with the fluoro compound. It's still a non-inhibitor of these 2 isoforms, and maybe putting this out here is going to make it a little bit more active in the primary target for fluoxetine. Now I'll switch back to the PowerPoint presentation. This slide shows our substrate classification models for various CYPs. As I stated before, we have 9 classification models for the major forms of, or the major CYP drug metabolizing enzymes. The data sources come from various sources. The major source here is the Accelerus metabolite database. We also use Drug Bank, a couple publications, one from Rendick and Sheridan, and then we always go back and look at original literature references. So again, as you see, the concordance and sensitivity and specificity for the test sets are all very nice, and similarly, or excuse me, for the training sets, and similarly for the test sets. So just going back into ADMET Predictor, I have a tab here for cytochrome P450 substrates. and it just shows yes and no predictions. One thing that I want to point out is that all of these compounds are antidepressants. Antidepressants typically are going to have a protonatable amine and an aromatic center, and that is also a formula for among other things, cytochrome P450 2D6. So if I pop into the histogram tabs, go to the categorical display, and then click on the 2D6, it's predicted all of these compounds are substrates for 2D6. In this next section, I'll discuss our sites of metabolism models. This was introduced in two releases ago in ADMET Predictor. This slide shows typical CYP450 reactions. The first example here is of carbon hydroxylation. So an oxygen atom is added between carbon and a hydrogen to form an alcohol. This carbon can either be an alkyl carbon or an aromatic carbon. Heteroatom dealkylation is formed from, again, hydrolyzing the carbon atom next to the heteroatom, where the X can be O, N, or thiol. This would create, again, an alcohol, but because of the X group, this is unstable. So, for example, if X is a nitrogen, this compound would be an aminal, which is unstable and would fall apart into the ketone or aldehyde and the amine. Another example of a reaction is heteroatom oxidation. So in this case, X is a nitrogen or a sulfur, and that can simply be oxidized to create the N-oxide or the sulfone for sulfur. And then the final example here is epoxidation. So a double bond or an aromatic bond gets converted into an epoxide. Currently, we do not treat these type of reactions in our models. So our approach to creating site-specific models is first to collect and curate data from various sources. One thing we want to emphasize here is the amount of curation that occurs, and I'll show a couple examples where we had to go back to the original references and really scrutinize the details in there. So again, we use the Accelerus Metabolite database and Sheridan as our experimental results. Then we classify the compounds as either, or the atoms of each molecule as either being metabolized or not metabolized based on the data in the first step here. Then we generate atomic descriptors for each atom, build neural network ensemble models that predict for each atom in the molecule whether it's a site of metabolism or not. And again, we built 9 models for this. And then the output of the program is a score for each atom in the molecule. And we would then take a look at these scores and automatically classify the highest scoring atoms as sites of metabolism. One of the important things to create good models for these sites are atomic descriptors. We've coded up a lot of these descriptors. I think we have over 150 atomic descriptors, and these are separate from the over 300 molecular descriptors. So we had a grant where we basically took a large number of molecules and calculated, performed ab initio calculations to create partial sigma and pi charges on the atom. So this is basically our experimental results, and then we parameterized an EEM and Huckel model to reproduce those ab initio charges. We also, from that, can create reactivity descriptors such as Fukui indices. Some of the other descriptors are E-state indices, local shape descriptors, including Sheridan's span descriptor. This descriptor is a measure of where the molecule is or where the atom is relative to all the other atoms in the molecule. So it would specify if a molecule or an atom is on the end of the molecule or if it's in the very middle of the molecule. And this would also reflect on how it would bind in the receptor and if it's available to get down to the iron group. And then we have other descriptors such as polarizability, electronegativity, or autocorrelation vectors. This is an example of a 2D6 model in one of the molecules that would go into the training set or into the data set. So experimentally, propranolol has 3 sites of metabolism as illustrated over here on the left. So there are 2 sites on the naphthyl group, the 4 position and the 5 position. Then the carbon in the isopropyl group gets hydroxylated and decomposes into the amine here. So our input to the model, we would have this particular molecule, and then the sites in gray would be specified as non-sites as input to the model, and then the red ones would be the sites of metabolism going into the model. So here, instead of each row in the spreadsheet being a molecule, each row in the spreadsheet is an atom in the different molecules in the data set. Then we start training the data set. So this is an example of the grid of architectures and then a truth table or confusion matrix for one of the particular architectures. As you can see here, this is a very nice model. There are 4,000 points in the training set and 546 in the test set. And as you can see, it's an unbalanced data set. So most of the atoms in a molecule are non-sites. They're not metabolized by cytochrome P450. And in this case, we've got a ratio of around 90 to 10%. And as you can see, we have very few false negatives, but a little bit more false positives. Not necessarily a bad thing to have some of the false positives, as we'll illustrate on the next slides. Okay, this slide illustrates our model performance for the various isoforms of cytochrome P450. So we have excluded and included atoms. So I didn't mention earlier is that if a carbon atom contains no hydrogen, then it can't possibly be a site. So we automatically exclude those from the models. And then here are the included atoms. These will be both potential substrate sites or sites and non-sites. So as you see, we have really excellent concordance, all above 85%, very nice sensitivity and specificity, and similarly for the test set over on the right. So another way that's been shown in the literature or how to compare results of metabolism is to tabulate the percent of molecules possessing an experimental site of metabolism among its top N scoring atoms. For example, if N is 2, then a molecule is counted as positive if one of its top 2 scoring atoms is a metabolic site, and that's why these percentages increase from going left to right. So as you can see over here, typically all of our models, the one side of metabolism is in our top 3 scoring sites. This next slide illustrates how we try to avoid overtraining, and this molecule is dextromethorphan. Experimentally, there are 3 sites of metabolism that have been reported and are in this SIMX Accelerus database. One site is the methyl group on the ether group. The next one is a CH in the cyclohexyl ring, and then finally methyl group on the piperidine off of the piperidine nitrogen. Our model, so here are the results from our model. So we give a score of, or predict a score of 985 for the ether methyl group and a score of 971 for the nitrogen on the off of the pyridine ring. So these are very high scores, and what you see is that the score for this other site is very low. So did we get this wrong? Is this truly a false negative? So if you go back and look at the literature for this particular compound, it was published in Drug Metabolism and Disposition and in 2009. And if you look more specifically at the publication, they were not looking at native cytochrome P450. They were looking at a mutant form called CYP2D6.49 that is forming this. So in fact, native 2D6 would not produce this product. So in fact, this is a true negative. Another example is chlorpromazine. In 2000, a paper came out discussing the sites of metabolism for 1A2 and showed that this is a site of metabolism on this phenyl ring. Our predictions show 5 sites of metabolism, so we correctly predict this site. We also predict the sulfur gets oxidized, hydroxylation of the carbon in the aromatic ring, and then these 2 atoms are degenerate, so they get the same score, but we're predicting these as sites too. So once again, we ask the question: is the model getting this wrong? Are these really false positives? Then if you look in an article produced in 2010, they actually, in fact, do show that these are sites of metabolism for this particular molecule. So false positives predicted by the model in 2009 became true positives in 2010. And then this one remains as a potential false positive. So let me just go back into ADMET Predictor and illustrate some of these sites of metabolism. Here I'm going to read in a new data set that was from FDA-approved drugs. So again, I go and calculate this. So these were drugs that were approved last year, so they're all fairly new molecules. And again, we can go into the substrate predictions and look at some of the substrate results, pop back into the metabolism model, and I'm going to go down to 3A4 and simply click on one of the molecules. So this would display the molecules and its scores for this. I can go through the various site molecules in the data set by clicking on this arrow here. Let me pop back to one of these that's illustrated in gray. So our model predicted that this compound is not a substrate for 3A4. And so even though we calculate the scores for this molecule, because we predict that it's not a substrate, we highlight this particular site in gray. Now, we do this so that if you know that this is actually a substrate for 3A4, you can still get the score for that particular molecule and the site. So let's look a little bit more closely at this compound juxtapied. I'm going to bring this into MedChem Studio again. And once you have it in MedChem Studio, you can simply click on the M button, and that will show the metabolites for this compound. We also allow you to control which metabolites you want to see. So I've only clicked on 3A4. I looked up the data sheet for this compound, and it is a 3A4 substrate. So let's see if our model correctly predicts that. And so it does produce metabolites from this on this compound. So one of the sites is this carbon next to the nitrogen, gets hydroxylated, and then again this forms an aminal, and it decomposes into the aldehyde and the pyridine. Again, this from the data sheet indicates that this is a true metabolite. We can also look at secondary metabolites. So this is metabolites of metabolites. So in this case, first thing we have to do is ungroup these two molecules, and then I'm going to move this one up a little bit and highlight it, click on the M button again, and it predicts sites of metabolism for this molecule and their metabolites. In this case, the carbon next to the nitrogen in the ring gets hydroxylated. Again, an aminal is formed, which would fall apart into the aldehyde and the amine. So this is a ring opening step, and was also mentioned as one of the metabolites in this compound. Okay. Going back into our PowerPoint slides, in this next section I'll discuss the CYP kinetic models, and these were developed in a similar vein as the sites of metabolism. So once we have the sites predicted, then the next stage is to predict the kinetic parameters for each of the sites identified in the site model. A couple background slides on enzyme kinetics. So you would have an enzyme and a substrate, would form an enzyme substrate complex. Excuse me, for the forward reaction, there's a rate constant K1, and the going back, a rate constant of K minus 1. Then the molecule would, or the enzyme would modify the substrate and then release the products, and this is in the in the second step where the enzyme substrate complex is broke, and that has a reaction constant of K2 here. This next slide illustrates Michaelis-Menten kinetics. So it's illustrated by this graph over on the left, which is a plot of the reaction rate or velocity versus the substrate concentration. So at each substrate concentration, you measure the reaction rate and then plot this as shown here in the red. As the protein becomes saturated at very high concentrations of the substrate, we reach an asymptotic level, and this is the highest reaction rate, so the Vmax. Then if you go down to half of the Vmax, cut over on the graph, and then down, this is the Michaelis constant, or Km. So Km is a measure of the affinity of the substrate for the enzyme. It's a property of the enzyme substrate complex, and doesn't depend upon the concentration of the enzyme and the substrate. Next we have the maximum velocity, Vmax, so this is the maximum rate at a fixed enzyme concentration. And then finally the clearance, so this is how fast the molecule, or the rate of clearance per milligram of microsomal CYP, and it is simply the Vmax divided by the Km. Again, going back to our propranolol example, for each one of the reactions with cytochrome 2D6, journal article has measured the Km of the various appearance of the products, Vmax, and clearance. So when we create our models, let's say the Km model, we would have these particular atoms that are sites, known sites, would be input to the model building procedure and would have the Kms for the various sites there. So why would someone use these kinetic models? So basically what you're trying to do is find evidence of where I should place my bets or what experiments to perform. It can be used in virtual screening where you have a large number of molecules and you want to avoid ones that are cleared rapidly. So if the model predicts very high clearance, then you should probably pick another molecule. Hopefully you've got enough virtual molecules there that you wind up in something in the end. And then the other thing is which assays to perform first. So if we predict high clearance for a particular CYP, then it's a good idea to have that molecule test against that recombinant CYP. This slide illustrates, again, our model building procedure for the kinetic properties. Again, we relied as a starting point the Accelerus database. Then we went back and carefully examined the original articles. And this was a very complicated procedure. A lot of the, especially in the 3A4 areas, if you're dealing with recombinant CYPs, the amount of cytochrome B5 and NADPH that is used in the experiment can vary from lab to lab, and you see various results for some of these experiments in the literature. So we also created models based on human liver microsome data, and in this case the amount of cytochrome B5 and NADPH is constant, so the variability from lab to lab is less, or from journal article to journal article. So we have three properties we measured for 6 different systems, so we have a total of 18 models. In this case, we used atomic and molecular descriptors in the procedure, whereas in the site's metabolism we were only using atomic descriptors, and then we developed regression models for each atomic site on the molecule. This slide shows the results of our KM model for 2D6. So here we have the observed values on the y-axis and the predicted values on the x-axis. The data set contains about roughly 100 atomic sites, and again we get pretty decent statistics with an R-squared of 0.8. This next slide shows the similar graph for the Vmax prediction, or the Vmax model, and then finally the clearance model for these compounds. And again we get a nice R-squared. So let's go back to Admap Predictor and quickly illustrate some of the examples here. So we know that Justipid is a 3A4, and then if we simply go down to the clearance models here, it shows that the clearance rate here for the recombinant CYP is 342. We click on that site, and that gray box, up comes the measured scores for this compound. So this carbon next to the pyridine nitrogen has a predicted clearance of 275. So this is a really fast rate of reaction. So again, we can go down through some of the other ones and look at the results. Again, if they're in gray, that's a non-site for the molecule. So those are all of those models. Now, going back to the presentation, we have a customizable score called a CYP risk score, which you can modify as yourself. There are default rules. So it indicates each molecule is scored as a liability, similar to Lipinski type rules. So if a compound has a particular CYP liability or it inhibits CYP, it's given a point, let's say. So the default rules are if the predicted clearance is greater than 40 microliters per minute per milligram of microsomal CYP for the 4 out of the 5 major CYPs. We also look at 3A4 and recombinant and HLMs, and again the cutoff is 40, but it has to be for both of these. And then finally we have inhibition models would get a score. So if it indicates that it's a particular inhibitor of any of the CYPs, it would have an additional score. I can just quickly show those in Admap Predictor again. So in the end of the Metabolism module, we have various CYP scores for all the molecules in the data set. I'll simply sort these in descending order here. So you see that this particular molecule here has a score of 3. It has predicted to have rapid clearance in 2C19 and 2D6, and is also an inhibitor of 3A4 when testosterone is the substrate. So in this last section, I just want to briefly introduce our UGT models. So a UGT is typically involved in phase 2 metabolism, and the enzyme transfers glucuronic acid onto the substrate molecule. So in this case, we have an aniline, and the UGT would add this glucuronic acid and make this molecule much more hydrophilic, and therefore could be quickly cleared for this. Now, most of the UGTs are present in the liver, but as I showed on an earlier slide, UGT1A10 is also present in the GI tract. So if molecules have sites of metabolism are UGT sites, so a molecule has to possess an NH or an OH group in order to add the glucuronic acid, then it can be metabolized by this. Typically this occurs in the second phase, so a molecule will be metabolized by a CYP, let's say, and be hydroxylated. Then that hydroxylation site can be glucuronic acid can be added to that via UGT. So again, just pop back into Admap Predictor and just illustrate again the yes or no's for is this a substrate of 1A1 or the various isoforms in of UGT. And again, we could plot a histogram of that. These are our 10 UGT models. Again, we have the number of compounds in the test set, in the training set, the number of total compounds in the test set, and then the various concordance, sensitivity, and specificity on the various models. Now, a model can also predict, so the results can either be yes, no, or the molecule simply has no sites for UGT. And then finally, just want to summarize, so roughly 75% of the top 200 drugs are metabolized by P450. Drug-drug interactions can occur when a CYP that metabolized one drug is inhibited by a co-administered drug. And then we have a number of models in Admap Predictor to predict these various properties, such as inhibition classification models, substrate, non-substrate classification models, sites of metabolism and metabolite visualization, atomic kinetic parameter models, and then finally UGT models for phase 2 metabolism. So I want to thank you, and I think I'll turn this over to Bob to address any questions.
Thank you, Michael. We did have a couple of questions we want to get to. There are a couple of questions about pertaining to how to make use of the numbers. One of which was, are the predictions species specific? And the related question is can it be used to predict the relative amounts of each metabolite and the overall metabolism by all the CYPs taken together?
Well, the answer to the first question is it's human. Our experiments are typically from human, either recombinant CYPs or liver microsomes, human liver microsomes. And maybe, Bob, you're the best person to answer the questions on the relative amounts.
The relative amounts, we're hoping to get it so that we are making good predictions for rank ordering. The situation is complicated for CYPs because many compounds are inhibitors as well as substrates, and so they typically do not follow simple Michaelis-Menten parameters. So having it so that they add up exactly is often not going to be the case. We're in the process of collecting data from very recent literature that we didn't use to build the models to help us try to help us validate exactly how predictive that's going to be. So far, what we've seen is it actually does a pretty good job of predicting the relative metabolites. Basically, as good a job as doing the in vitro assays does that we used. That's where the data came from that we used to train it. As the reader, as the listeners may know, there's quite a lot of variation from lab to lab in the exact amounts of different metabolites you see, depending on exactly how long you leave the reaction going.
Very good.
Another question we had was about when you're showing a graphic of the molecule with some of the atoms highlighted in red, there are numbers attached to those atoms. What do those numbers mean?
Those are the scores for the site models. The higher the score, the more likely that site is. And what we do is look at all of the scores for the atom and then highlight the ones in red that are the highest out of all of the atoms. So a certain percent. If all the scores are very low, we might have a cutoff and indicate that there's simply no sites. Our scores are very low for that compound.
Okay. And another question, which is sort of general, and it wasn't actually clear to me. It's not clear to me whether it's about the experimental data we used or that people would use to make their own models. What experimental data do you need to provide input for your model building?
I'm sorry, can you repeat that question?
What kind and how much experimental data do you need to input to the model?
Okay, for the substrate non-substrates, is this a substrate for that particular isoform? For the Km Vmax models, it was the measured Km for that particular reaction. So it wasn't on the overall molecule. We had to identify a particular product for that.
We have two more questions. One is, do we plan to include induction prediction in the future? And I'll answer that one, which is that somewhere in the future, yes, we hope to. There isn't a lot of good hard data on that yet.
Yeah, and I didn't cover induction in this also, but so we have compounds that are substrates for certain CYPs and are metabolized. We have other sets of compounds that are inhibitors of cytochrome P450, and that can lead to the buildup of another molecule. Also, some molecules cause inductions of CYP, and so they increase the clearance of a particular molecule. So if one molecule is an inducer and it's co-administered with another molecule that needs that CYP or as a substrate for that CYP, then that other molecule will be highly metabolized and therefore may not reach therapeutic levels. And so if you look at these data sheets on various drugs, it's going to say what the compound potentially is if it's an inducer, and in the case of if it's an inducer and co-administered with a substrate for that particular CYP, then you would increase the dose of the other compound in order to reach the therapeutic level. So typically the data sheets will say, okay, if you're co-administering this with an inhibitor, then you need to reduce the dose of this compound. If you're co-administering it with an inducer, then you need to increase the dose.
And I think we have time for two more questions. One is, why do we not provide a model that includes epoxidation? for the SIPs?
I can answer that. There's not enough data of sufficient quality. That question was asked by John. John, if you have enough data on epoxidation in your company and you want to share it with us, we'll be very happy to build custom model for you, and you'll have it.
The other complication there is that the most of our models are built on atomic properties, and what determines epoxidation is a bond property rather than a single atom, because it's taking place on a double bond and not by hydrogen abstraction from an atom. So it's a fundamentally different kind of process. And the last question I want to address here, well, one is, will the slides be available? Yes, we will send information to you. We will broadcast information about how to download the slides. And the other was about whether the new licensing system was going to be available for the next releases of our other software. It will be available for the next release of MedChem Studio. It is not going to be used in the next release of Gastroplus because the timing didn't exactly work out. However, we do plan to have it in the release after the upcoming release of Gastroplus. And I think with that, we probably should wrap it up.
There's one more question
we didn't address. I'm sorry?
There's one more question that I can very quickly address. Do the models provide any indication of whether they are applicable to compounds or not? Yes, we have an indicator of a given compound being out of scope of the model. When Michael was showing predictions, in ADMET Predictor, these predictions were highlighted in magenta color, as opposed to black font that was for the compounds that are within the scope of the model. Also, some of the classification models now feature the probability of answering being correct, and Michael talked about it.
Thank you, Robert.
So we did want to turn it back over to Anu to kind of wrap things up.
Okay.
Go ahead, Anu.
All right. Thank you so much. On the screen, as you see, is our contact information. We offer 2 weeks of free trial license. If you're interested in evaluating the product or if you have any more questions, please feel free to call us. I am responsible for the ADMET Predictor in the MedChem Studio, so please reach out to me. My phone number and email is on the screen. And if you have any questions for Michael regarding the consulting services, the simulation and cheminformatics projects, then Michael can be reached at there's contact information on the screen. And if you would like to evaluate Gastroplus and DDD Plus and have any questions for that, please reach out to John DeBella. He's our Vice President for Sales and Marketing also. So I will be following up with you in your email if you need the recording of the webinar, we will be sending you the webinar recording as well as the slides, and we will be posting the same on our website as well. And next, April is going to be a very busy month for us, so we will be at the American Chemical Society meeting next week and the Drug Discovery Chemistry meeting. If you're attending, please don't forget us. Stop by our booth. We will be in the exhibition hall. And also, if you are a Gastroplus user and would like to learn more about the product, then we invite you to our Simulations Plus workshop in San Diego on April 22nd to 24. For that, the information for the registration is on our website, or you can reach any one of us and we'd be happy to register you for the same. So once again, we really appreciate your time and your attention, and we appreciate your interest in our products, and we look forward to serving your needs. So please feel free to contact us anytime with any questions or queries. Thank you so much.
Thank you, everyone, and bye.
Bye-bye.